Yes, happy Friday. It's been quite a week, but next week's a two-day week, so hopefully we just have to push through until Thanksgiving. Today my video is going to be quickly about DNA replication, which is part of the cell cycle you guys are learning. Remember, it's G1, S, G2, mitosis, and then cytokinesis. And during the S phase, during interphase, DNA replication occurs. There are 4 steps to DNA replication that you will need to know. The first step is just the normal DNA. So it starts with a parent DNA strand. As soon as the cell enters S phase, it's a double helix, 2 strands held together by hydrogen bonds. Oops, I spelled together wrong. Then what happens first is DNA helicase breaks the bonds between the 2 parent chains of DNA. So DNA helicase, just like during protein synthesis, DNA helicase breaks the hydrogen bonds between the 2 chains. Once the 2 chains are broken apart, the third step is that DNA polymerase builds the 2 new strands of DNA. You can see the 2 new strands here in green on both sides, and the enzyme DNA polymerase builds them. This is a little different than protein synthesis, because in protein synthesis we are building RNA, and so it was RNA polymerase. This time it's DNA polymerase. And then what you end with is 2 identical molecules that are half old parent DNA and half new DNA. This will happen all the way up the strand, and so all the way going up this way, and it will split all the way through, and it will go all the way through the huge long strand of DNA in every chromosome until you have 2 complete sets of DNA. Then I just wanted to show you this slide to highlight each strand is half old, half new. If the parent strand is shown as in red here, when it splits, the 2 strands, new strands are in purple. They're still identical to each other, so TA, AT, GC, GC. So they're identical strands at the end, but each has half old, half new. That's all you need to know about DNA replication. I will see you on Tuesday. Be sure to watch on Monday. You'll have another video about cancer.